All right, thanks everybody for joining today. This is our first DNA workshop where we're going to be studying basically mutation data fundamentals and visualizations. And this is just an extension of the framework that we've already built in the course of our 15-part workshops, which really started you from the beginning and worked you up to kind of proficient level to be able to understand, just fundamental theory of data science, right? And we're working within the realm of genomics data, right? But this can be, those first 15 lessons can be applied to any data science lesson. But now for this semester, we are going to move on and basically apply these techniques. So far we've been working on more of the theory, right? So now we're going to do more of the applied. And here we're going to do it in the realm of genomics. It's going to be really applied genomics. We're going to go through several DNA workshops. We're going to study copy number, RNA, RNA-seq, things like that. As we move on, it's going to be really great. So thanks everybody for joining today. Glad to have you back after those crazy 15 starting videos, right? Let's move forward on this now. So, silly question, obviously a lot of you know this, maybe some you don't, but why are we studying mutations in cancer? How could that be important? Well, come on, it's pretty obvious, right? Over time periods, your body builds up a lot of different mutations based on age, based on environment, based on genetics, right? And, depending on what the mutation is, it can cause, benign effects, or it can lead all the way to being a driver for cancer, right? So obviously, in this realm of big data, we can look at all the mutations and start understanding more about what each patient issues are, so we can develop, personalized therapies. And you can see here, we can, a lot of therapies MD Anderson developed, targeted therapies with EGFR, the big deal, immunotherapy, right? If a cancer can have a strong immune response, and we can see that with the mutations, then we might be able to show that, immunotherapy is the right treatment in this case, right? And really one of the great benefits of having these mutations is really understanding what's going on really early on. Early detection, early prognosis, you can be able to treat cancer correctly. But remember, all we're going to be studying today is basically just text files, right? Or Excel files, right? It's just a formatted file. So even though this is genomic data, it can apply to anything, right? Some of the packages maybe don't, but the idea, all we're doing is using just simple data frames, simple matrices, and we're just reorganizing that data. So I'm going to show you several different file, formats. It's the same data, it's just formatted a little bit different, which is really the hardest part of data science and computational biology. It's just formatting the data right and then knowing what package to use. So today we're going to be studying, our cancer model we'll be working this today will be the breast cancer model, and we're going to be using the TCGA, the Texas Cancer Genome Atlas. Why not? It was partly established MD Anderson, and it's just a great, catalog of thousands of cases where you can understand, really get a look at bulk mutation data really easy. And just to give you kind of an idea of what we're hoping to look for after our analysis, there's 5 different main subtypes of breast cancer, luminal A, B, HER2+, basal, normal-like. But what's interesting is these are wildly separated mutations. So if you know the mutational profiles of your patients, you can get a really good idea of what subtype they are, which obviously is going to help their treatment, right? So what are we expecting to find with this data today? Well, we're expecting to see that PIK3CA will be our most frequently mutated oncogene. We're expecting to see TP53 up there pretty high too, right? But also enriched in more of the aggressive subtypes. And we're also going to see CDH1 mutations in, for example, the lobular carcinomas, right? And we're expecting a low mutation level, low mutation burden overall compared to other cancers, and really diverse heterogeneous mutations here. So let's jump in and let's check the data out and see what we got, right? So by the end of today, I really want you to kind of understand how to load and navigate basic math files, mutation annotation format files, using the Math Tools package. Kind of really understand what variant annotation concepts are, and how we annotate and understand functional mutation predictions, and just be able to kind of calculate some descriptive statistics. We have mutation data, like how many TP3 mutations are there, how many of this, right? Just look at the counts, things like that, right? And then we're going to also analyze some statistical distributions within the while we're looking at the tumor mutation burden to kind of understand a little bit more how the data is just how it looks, how the data is organized. And then we're also going to create some really cool publication quality visualizations that come right out those math files, and you'll have the data right there, and you can apply that to your data as well too. And we'll do all this within the context of this breast cancer, right? So here's kind of some packages that I showed you to install. Just like last semester, if you need to install, BioC Manager first, and then install all the bioconductor packages by the Bio Manager, install like I said before, and CRAN package just like normal as well too. Most of these you should already have, but here are all the ones that I loaded here today, and kind of a description of each one of them you can kind of check out later. And let's see, let's just jump in, right? So you understand TCGA, right? It's just basically they just built up this landmark of cancer genomics program that has analyzed 20,000 tumors across 33 cancer types, and you can pretty much download the data. But the problem is it's really hard to get the raw data. That's kind of closed door. You have to send a lot of emails and ask for a lot of stuff. So we're kind of going just a little past the raw, just a little bit normalized, or just a little bit, mutations are great because they don't need to be normalized. So we're just getting a little bit past that. And I'll explain that a little bit. So today we're going to download what's called the masked somatic mutation data. So don't worry too much about what that means. It's the mutations. So the mutations have been called by really TCGA, usually MUTEC. MUTEC 2 is a newer one, but the old MUTEC probably. But there's different algorithms that can call mutations, and that's basically what we have today, right? So basically they've called mutations, they've annotated the mutations, they've given us to a final file basically that has all the information. And that's great. We can work with that today, but how did they get that? You're not always going to have that. So I'll also show you how to get there too, right? So just like last semester, you can kind of check back at some of those, how we downloaded the TCGA data. There's lots of ways you could do it. You can go to C Bioportal and download some of it, but it's really hard to get the right data. So it's really better just to query it and ask for exactly what you need, right? So this GDC query, just like last semester, I got my project. Now we have a different data category, a different data type than before, and a specific workflow, right? And this is just something you're gonna see that when I make mistakes like that. Q, cut, and change. So you'll have workflow types, and you can kind of search these. ChatGPT, Claude can help you, or looking on the GDC websites and such. You can get these kind of exact things you need. There's very specific things you're asking for, right? And then you kind of look at the available samples, and you do a download based on that. And just like last semester, look in those videos we did for statistics and machine learning. One difference here, we have to kind of read in these math files. It's a little bit complicated here, but I've done it for you. And feel free to copy this code later and just use it for yours. But you're just basically just pulling in the downloaded data and just kind of organizing the file, changing it to numeric, things like that, and getting the final file. And you can see it is the final math file rolled in is a BRCA math file, right? And before that, we had math data frame clean. So let's check this clean file out and see what's going on with that. So what? Math data frame clean. Math data frame clean. All right, let's check this out. So yeah, so really it looks like we just organized it, right? We just have our gene symbol, where it was sequenced, our build, which is really important because you need to have the right coordinates, right? And here we go, for each gene that has a mutation, it has the chromosome it's on, start position, end position, the strand. You have all that good information. Really all the information you need that came out of that data you're going to see in that math file, right? But this is kind of the clean version. It's still like a data frame, right? And that's great too. We can work with that too, but we want to put it into this math file, right? So when we usually make that, let's see, it was the BRAF math, right? Where is that guy? BRAF math. The final one you can see is basically they put into a different list here. So if you look at here under data, and if you ever want to open like one of these deals, you just go over here on the right and you can click here and that kind of just opens it up back. And this is exactly, so the BRAF at data is exactly what the BRAF clean file is, right? And it just kind of did some more stuff and organized, kind of made some counts for us, made some summaries for us. And if we had the clinical data, which we won't do today, it'll pull it in here as well. We had a couple here, but this is the clinical data I actually added later. So this wasn't on here later. Should have jumped to that. But really, all the summaries things, just a way, it's just organizing data, right? Like I said, we just have the data and it's organizing it in a way where we can use it an easier way for packages. So that's what you got here. The math data from clean is really just all the data. The BSRV math, after I did the read.math function, it gave us that function, right? So usually sometimes if you do your own data, you would have a VCF file first, right? And then you would work on, I'll talk about that in a minute, you'd work on and do some different things to it to get to your math file. Well, we don't have a VCF file. You can't download the VCF file without getting past the paywall, or not paywall, but just defense wall, I call it. And it won't be it's hard for you to recreate that. So I wanted to just not take us that far and just basically reverse engineer a VCF file for us, just so we can learn, and just for a learning experience, right? So don't worry too much about what I'm doing here. You can check it out if you want. But all I'm doing is reverse engineering that clean file back into a VCF file, right? So I've done that. I've added a header, and I've basically written the file down. And now you'll see we have a couple of different things here. We have our BR-CRV. So what I did was I made the object, right? BCRA VCF, right? So BCRA VCF, and I made the file, and then I basically created the file, and then I pulled it in with read VCF to make a VCF object. And so you see the VA. And I'm just saying basically the VCF, it's pulled in, VCR, VCF, VA. Let's check that out. And here we go. Once again, it's kind of pulled out, kind of like we had before. It's different things in the list. You have the ranges of the different the different mutations you see here. If you look here, the different mutations. It's just it's the same data. It's just reorganized. Let's check out the VCR of body. That's probably the best way. When I made the file before, I kind of made it into the object, if you remember. So here's we go, VCF body. So it's the same stuff. Look, so here's what a VCF file would look like, basically. If your chromosome position, kind of the same stuff, right? What the alternate allele, we'll look at that again in a second. But that's the VCF file that I've turned into the object, right? Here to use later. And then I've also made it into a G-ranges object. You would just use that VCF file that you read in with our VCF, and you do row ranges on that, and it grabs the G-ranges object. And really all it's doing is grabbing, if you look here, this row ranges object and pulling it out. But anyway, the idea is we would have math files are readily available like that. You can work with them. If you don't, you can start with your VCF file. This one, just think about it as a teaching tool that I've kind of gone back and reverse engineered for you. So first, let's understand, these different mutation formats. Just kind of stick with me here. It's going to be a little dense. And then once we get through the formats and the annotations, then we'll move on to the code and some cool plots, things we can do, right? So first, the math file format. This is kind of the high boss file, right? That has all the information. So this is just basically the standard format for somatic mutation data that everyone uses. And think of it as like a very, very detailed spreadsheet that we've seen, where each row represents a sequence mutation and the columns represent different observations, right? So let's just take a peek at that. Well, look, we did before, right? Remember the BRAF math data file? And we just kind of grabbed some of the columns, took the first 10 rows, just like I showed you before. You already saw we have the mutations, chromosomes, start in positions, the reference IL. What's the change? Well, there should be a C, there's a mutation, now we got an A. So it's the tumor allele change, right? And here's kind of the variant. We'll talk about that a little bit, and the protein here, different changes. And if you kind of go through these here, better to see, probably should have just gone here first. But Hugo symbol, your gene name, chromosome number, start in position of your mutation. If you have a single point mutation, they're going to be exactly the same number as you see there. And here we got a C changed to a T. We have a variant classification, because you remember that math files are already annotated. So you already know if they're a missense, and you already know the protein change. You're already going to know how deleterious mutation is as well. So let's go back to the VCF format. So what if you only had the VCF file, right? It's just the same thing, but it's simpler and more universal, right? Has the very basic information, and where the math file kind of just added more stuff to it and organized it in a specific way. So a VCF file is really for any genetic variant, the very minimal you need, just the variants. And it's smaller and compact, and it's based on just the very beginning of what the mutations, once they've called the mutations, that's all you got, just a very basic. Where in contrast, the math file, remember, is very highly annotated. It's looking at cancer notations only, and it's just is the next step, it's just higher annotation, right? So VCF is the raw ingredients I said here, and MAF is the full meal, right? So like the raw ingredients could have done a lot of different things, but the flavors are just cancer. There's my silly annotation. So, and we looked at the VCF file. If you remember, we kind of checked out and we saw those kind of easy. We kind of looked at the, as here we saw just specific mutations. So you see this is just the VCF file like we showed above. There's the reference allele, the changed allele, and we have our specific location, right? So instead of having the gene names, we just have the IDs, right? We don't have the gene names yet. We just know chromosome 1, this location, T to C. So remember, that's very general. So the math would tell you the mutation and then also tell you more, right? If it's a missense, on and so forth, right? So this is just a very raw data, very raw data. And you see here these RSs, these are normally SNPs that have been called. So they're like mutations that are highly seen at a high frequency in the population, so they're probably unlikely to be dangerous. You can still check them out later specifically, but these are the kind of things that are going to get cleaned out when you make that math file. It's going to take out low variants and SNPs, things like that. That's why it's so highly annotated. But the VCF's great because you have all the raw information, right? And where you can go any way you choose. So it's kind of the same way as before, chromosome, position, your ID, reference allele, alt allele. One thing you will see added a little bit on the end, you'll see things like maybe quality and filter, and that'll kind of just tell you, depending on what kind of run you did, it'll let you know how successful the sequencing run was. So if it has a horrible score, maybe you can't trust this mutation, right? So basically it's saying at this mutation, how well do we believe it? It might give you the allele frequency here or the depth of reads. These are all things that make you have more belief in that being true. And finally, a lot of times you'll get this final thing, the sample genotype, and basically that's going to tell you if it's heterogeneous or homozygous, things like that. So 0 to 0 is homozygous reference, so no mutation. 0 to 1 is usually one copy of the variant, heterozygous mutation, and then homozygous you usually get both. So that's what you're going to kind of expect from that. And VCFs come with a header. It's just kind of important. They tell you kind of where it came from, how it was created, things like that. Those are kind of always associated. So really you'll get that body file that I showed you. That's basically the VCF file, just kind of tells you, where it comes from. One final object is not quite as important, depends on how you use it, but basically bioconductor is a very robust way of analyzing a lot of genomic data, and it has tons of packages. And one way you can make your data more applicable or easy to use for all of the different kind of packages they have is to make a G-range object. And if you remember, that just comes out of the object of the VCF of that math or the VCF file we have, right? It just wants the ranges that are a little more detail, right? And just cleans up a couple little things. So let's kind of take a look at this object. If we see here, what's the best way to look at this? We'll get it down here, I think. I made a preview of it right here. And you see here, chromosome name, start, end. So we have where the mutation is. It has our reference alleles. And we also have strand. It has we have a dot here because I didn't pull the information off the VCF file. But it would be like plus or minus, which DNA strand is a mutation on. And this is just part of the G-range object. It's a very clean object. And the width of the mutation is very important too, like an insertion is 24 base pairs, things like that. And this just, like I said, it puts it in a better format to be used for bioconductor packages. So once again, sequence names, chromosome, number, start and end, the width, the strand name, the reference alleles, really the same stuff, right? It's almost the same thing. It just likes this file a little bit better. So just kind of knowing that, good to know because you may come across that, right? So remember, VCF is a universal, very, very basic information, tells you what it is. Math has more detailed annotations and is more cancer specific. It tells you more about what happens with those mutations that VCF found, right? And G-range is kind of like a souped up VCF that just fits the bioconductor, data pipeline a little bit better. So now you know a little bit about the overview of those files, and this is how they kind of go together in a basic cancer genomics workflow. So basically, when you do a sequencing run, let's say we're doing mutations, you get all the data, and it's really a big mess and complicated. And the first thing you do is you demultiplex those files. You just basically put the data back into the FASTQ files, and that's basically what you guys always see, right? A FASTQ file. And that just gives you all the base pair changes and all everything that the different reads saw that seem different, right? But they're not really aligned to our genomes. The first thing you do is create this BAM file. You create a SAM file, then a BAM file, but just to keep it simple, create a BAM file It's just aligning those reads to the human genome. And that's why you have to know like which build you're on, like the 38 or HG19, HG, you need to know which build you're on so you'll have the right genome to align to. So now you have all your mutations aligned actually to the actual genome, and then they'll give you a bed file. Don't worry about that one. And then you change that into a VCF file. So these are just kind of the main stepping stones here. Then you have our VCF file, just like we talked about, right? It has the basic mutations, but nothing more. And then we make that into an object, and then be able to change that into the G-range or the math file, and be able to do that to do that's where we do our downstream analysis, do our annotations, everything like that. So it's just kind of the sequence they go through, just so you have a little bit of understanding of how that works. So why do we need to understand variant classification? So what does that even mean? Well, so just because you have a mutation doesn't necessarily mean it's bad. Some are not even a big deal at all. Some are better. A lot of them are worse, depending on where you get it. So depending on what a mutation is, how do we know, or how can we find out, like how does that relate to the how dangerous and pathogenic the mutation is, right? So just if you guys don't know this, just to kind of look at some different classifications of mutations, you have missense mutations, which is just one single change, like C to T. We've seen those, and that can change the protein sometimes. Sometimes it doesn't. And that's just a moderate bad. It's not horrible, right? But the bad ones really are the ones that really change the proteins. You have a nonsense mutation. That's premature stop codons. That truncates the the protein doesn't get fully built, right? So frame shift. You're just causing major frame shifts. So you might take 10 different base pairs and just rip that out and move it over somewhere else. That's going to change the protein a lot. You know what I mean? Or what about RNA splicing? Or what about, insertions? So, indels, insertions, deletions, this can really change how the protein works. That's why the impact is so high. Or something like a missense mutation, an in-frame deletion, so it doesn't change the frame, a loss of the stop codon. Well, it's not the end of the world. The protein pretty much got made. So those aren't that bad. And once again, the translation start, that would be a bad place because the protein wouldn't start to get built, right? So really just think about it. Mutations that mess up the protein the most are going to be the worst, impact, right? So this is just good to know. And this is the kind of things that we can annotate on the mutations. And this is what these databases are for, right? So how do we know? How do we do this, right? So when we have that VCF file, how can we figure out, that tells us what the mutations are. How do we know what they do? And well, that's where annotation tools come in, right? And two of the most famous ones are ANNOVAR and VEP, right? And basically VEP is a variant effect predictor. ANNOVAR we don't need the whole name. Annotate variation, and if you want to learn a little about how they work, the ANNOVAR just takes a tab delimited text, so it has to be more like the VCF body. It can't be the list of objects, right? But the VP will take the objects. So you can take like that VCF file, you can put it in there straight up. And basically it's going to go through, it's going to look at each mutation, it's going to tell you what the gene is, and it's also going to give you your variant classification. And then it's also going to give you a statistical measure of how dangerous that is. And I will show you how this works. Now, the good news is the math file already has all of that. So if you could do what we did earlier, and you just download the math file first, then you already have all of this. You don't need to do anything more. But I just have the VCF. What do I do? I just have the VCF. Well, that's okay. You can run the ANNOVAR or VP first, and then you can convert that over to the math file pretty easily, and then you're good to go, right? So why can't we just change the VCF to math straight? Well, remember, it doesn't have the annotations, right? So math has everything, right? VCF just has what it is. It doesn't have the VCF has what it is. It doesn't have the gene symbols, the classifications, all the different things on there that you need. So that's why you can't just do it right away. You have to do the annotation first. Basically what you're doing is you're basically connecting to external databases, right? And just looking at things that have already been tracked, already been put in. And you're not doing a novel thing. You're just looking at what already has been done, using that information to annotate, pull that out of a database, and stick it in your file, right? All we're doing is building like an Excel file. And we're just adding more information to it and organizing it in a way we can use it. It doesn't have to be genomic data. It just happens to be in this instance. So just one more reminder, we have our raw VSCF file. It just has like the exact location. Then we run it something like VEP, for example. It looks through, different kind of protein sequences and different kind of big genome databases and decides what things are. And then it gives us our annotated math file. And look all the great stuff we have here. Now we have the mutation, the gene symbol name. We know it's a missense. We know it's deleterious, which we'll look at in a minute, and we know the protein change. So that's how you get from start to finish there. Now, how can you do this? Well, this is one issue that I'll have to say is that R is not a good place for this. So a lot of when you go from like FASTQ files to VCF, those are kind of high level things that are normally done on the command line. And they're usually done for you, to be honest with you. You usually get those files from the cores. But if you have to, I can work with you later. It's no big deal. It's actually really easy. You just have to kind of know a little bit of knowledge about how to install some of these programs or some of these or bring in and run some of these algorithms. So for example, there's a file called vcf2math.pl. It's a Perl file. You can download this and run it basically. So really, if you want to try, you can pull in that VCF file I have and just download this Perl file and just run it on the command line. You would just put the Perl file and you would pull in these flags for it, and you're basically just putting in your input VCF, what you want it to be called, and then the human genome. You have to download that as well too, and what path do you want to go to. And just this one command here will take your VCF on the command line and spit out your completely annotated math file. So really all you need is the Perl file, and if you really want to do that, if you have some VCF files, I'd be happy to work with you to show you how to do that. We're kind of we can't do that in R. We can, but it's just too complicated and it's not efficient, right? So if you want to do that later, just know that's how you would do it. But the good news is we don't have to do that today. We have our math file that's already annotated. So that's really good news because TCGA takes in the FASTQ file, already runs VP, creates the math, and gives us everything we need to do. So the only time you would need to do the annotation is if the core or somebody gave you a VCF file. And like we mentioned, it wouldn't be hard to move over and do that transformation over there. All right, so I showed you like how it told you if it was a missense or a nonsense, and then I kind of gave you like a hand-wavy reason why that might be bad or good, but how can we know like from a statistical standpoint, like a numeric standpoint, give it a score to know how bad it is, right? So the two best ways of doing this, or most famous, are SIFT and polyphen. And basically we want to know if you have the mutation, especially a missense with these two, how bad will this break the protein? How much will it mess it up, right? So SIFT, which is sorting intolerant from tolerant, basically uses evolutionary conservationism to predict the impact. So what it's doing is, it's basically saying if a base pair position is conserved very strongly across species, it's very likely it's very important, right? So if you mess this up, more bad things will happen. So it's really just looking at evolution and how well these are conserved. And if you kind of just basically looking in the BRAF data, and basically I'm just counting up how I'm looking in the SIFT category, and I'm just basically finding if they said deleterious or tolerated. So I'm basically just going to the BRAF file, the data matrix, and just looking in the SIFT column and looking to see how many of these are. I'm just doing a simple count here, simple count, and we're basically see that there's 31,000 deleterious mutations and 24,000 that are benign. And what does this mean? So basically, if you're less than or equal to 0.05 with a SIFT score, that amino acid is likely to affect protein function. And the lower the number, the more dangerous it is. And in contrast, if you're above 0.05, it's unlikely to change the function. So you could have just a mutation in DNA that leads to a protein that's unchanged. There's no difference. So who cares if you have a mutation? And that's how a lot of mutations are, really most. So that's how you kind of look at those scores to see how bad. And then the higher the number, the safer it is, right? Other one's polyphen. And this one uses evolutionary conservation too, but it also heavily relies on the actual protein structure. So I want to look, certain proteins are more, they can resist, change more, even if they have mutation. And it's also going to look like, where is that mutation on the structure? Because there's some parts of the protein where if you change the mutation, the whole thing could fall apart. Or it might not be able to link on to certain different things. So it really matters where it is in there as well too. So it's not just conservation. And I looked at the same thing, and this time I looked in the data for the polyphen category. Same thing, and looked at kind of what the different designations, and we see we have 25,000 benign, 19,000 possibly damaging. So like half of these would have been in this category for the CIF. So, and we see that 10,000 are possibly damaging, right? So you can kind of compare mutations back, and usually you can do things like say, I want a mutation that's less than 0.05 CIFT and then greater than 0.85 for polyphen. And if they do both of those, then you know it's bad, right? That's the kind of thing that I'll kind of look at. And these are the kind of, and I kind of skipped over, these are the kind of ranges of the scores you would look at to be able to classify those categories I just mentioned. And what's kind of annoying is high scores better for polyphen and worse for CIF. So sometimes I'll make a CIF score, I'll do 1 minus CIF for that score, just to be able to compare them a little bit better. Anyway, okay, so let's move on. So those are the two classical ways that people use, and you're going to see that a lot in papers. But there's a major problem with this, and I'm going to show you how I believe that CAD has a better pathogenicity score. So what is the limitation of CIFT and polyphen? Well, there's a humongous problem. It only works for coding variants. So the mutation has to be in the coding area, right? It ignores spice slides, regulatory reasons, other things that are really, really important. Nonsense mutations, stop codons, intront variants. These are all things that could be very, very, very important to mess up that protein's function, right? So even though the other tools, most missense mutations are found in coding regions, that's why those tools are so powerful, it doesn't catch them all. So what CAD does, this is a really cool, I forgot how long, maybe about 10 years ago it was made. I have the paper listed below. It basically solves this by scoring every single variant in the genome. It doesn't matter where it is. It looks at SNVs, indels, any variant in the entire genome, coding, non-coding, dark DNA, it doesn't care. And it uses 60 different annotations combined. It looks at conservation, regulatory, protein or epigenetic, blah, 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 all the way, 57 more, right? Instead of one thing or two things, it looks at 60 different ones, combines that all together into this great Phred score that is very easy to read, where if you're over 10, you're in the top 10%, over 20, you're in the top 1%, over 30, you're in the top 0.1%. And over 20 is usually considered pathogenic. And so this is just, I always love this tool. It's really, really cool because it just pulls in everything, gives you a much more detailed idea of the effect of mutation. And just to kind of go to that website I'll show you here, is University of Washington website here. And what you can do here is, here are the papers, you want to check them out. Click up here on score, and basically you can upload your VCF right in here. And you can also do, let's see, that's how you find, you can also go, if you want to just type one in at a time and just check one, you can, or you can do your whole VCF, or you can just go download. They've already pre-calculated this for everything. This is just the database. You could actually download the entire file. They've already calculated this for every single spot, or every possible mutation possible they've calculated already that we know about, right? So you can just do that and just look at your site number and just kind of just see what they are. But the easiest way to do it is really just putting your VCF file. But that's a pretty easy way to get all that, right? So let's see, I got pulled down here. Let's go back to where we were. Why would it take me away? All right, so we're on the CAD, right? So so this integrative approach, it kind of gets you past. So if you want to look at just missense only, so polyphen, it's already there, and the math already calculates this. So those ANOVAR, VET, all that stuff is already calculating the CIF polyphen scores, right? So that is kind of quicker. So you kind of have to just do one extra step if you want, but I think it's well worth it. And if you just look at some of those, if you want to look at some of the annotation data that I mentioned, you can, I basically pulled in the data here, all the column names, and I'm just looking at the CIF polyphen categories. So if you go back to the brow file, there'll be CIF polyphen, cosmic impact. Depending on what the database is, there's different columns within that math file that'll give you that information. For example, if I grab the data and I look at just three genes, and want to grab just a few things here. I want to grab the SIF, polythin, and impact. So I'm just looking in that BRAF data file once again, and I'm just pulling out, sorting out columns that I want, selecting columns just like you did in the first 2 weeks of the boot camp, and pulling those in. So here's kind of an example of what that file would look like. Just looking, kind of looks at some of the key mutations. This is just basically just selecting some. You see T53. We have the protein change, the missense, and we have deleterious and the score here. So this is 0, super low, right? 0.04, just right below the 0.05, not as bad. 0.01, pretty bad too. This one was probably the worst, right? And but even though they had a missense mutation, it's still a moderate impact because these kind of mutations don't have as high an impact, right, in general. And same with nonsense. We actually don't have the answer, right? Because they're not coding, right? So here's a perfect example where we need the CAD score here. We know they're high impact, but how high impact are they? This is where CAD could fill in these details for us. And you can there's other tools too. I just think CAD works pretty well. So going back to those SNPs I talked about earlier, those RS things, another thing is like how common is this mutation in healthy people? If it occurs a lot, it's probably not that big of a deal. Otherwise, we'd all be dead, right? So there's several different good databases here, like Genome AD, that aggregate all those SNPs for all of people, and you can go and check. And when you do that VEP analysis to make the math tools, it'll look at those, and if it finds ones that it knows aren't dangerous, it'll throw them out. So they'll be in your VCF, but they'll end up throwing out your math file, right? So let's look. So now let's just go through that. Now we come through how everything would look, and let's just move on with the downstream analysis a little bit and look at the math file. So now we have our math file. We have all our annotations. We've done all the things we need to do. Let's do our analysis. So the first thing we can do is just look at a summary, right? Just an easy summary. So if you do get sample summary or get gene summary, you can get likewise simpler descriptions, summaries of these objects. I'll show you what they look like. So what math and gene summary. So the math summary is right here. And you can see how it's basically just per sample, what is the distribution of different mutations? How many indels do you have, right? How many missense? 4,000 in this one patient, right? So there's 4,782 total mutations in this patient, right? It has all our patients. That's kind of the summary there. What if you want the gene level, right? So now we need the gene summary. So it's the same thing, exactly, right? We have our Hugo symbol. So for TP53, here's the distribution of mutations. There's 341 of them in the entire data set, right? So that gives us a couple of our summaries, and we can also do head of just the math summary here, just showing you the same thing here. Same stuff, gene summary. And we have, if you do print in the whole file, you get this kind of big overall summary of everything, which is nice too. So let's just kind of let's just do some descriptive statistics and just kind of count up what we're looking at, right? So number of samples, I'm just looking at the BRAF data, same thing we've looked at before, and I'm just looking at all the barcodes to see how many samples there are. And I'm calling that in samples. Then I'm saying, because I'm looking at unique, right? And then I looked at the number of rows completely gives us the number of mutations. But the number of unique gives us the actual number of samples because it's going to have a mutation and not every sample, right? So we don't want repeats of the barcodes, but we want all of our rows. And then we can look at our number of genes, kind of the same thing. I looked at unique for the Hugo symbol. So I'm just getting unique genes, unique tumor IDs, and all the rows and mutations. And if I plot those out here, you see we have 990 total patients, 67,000 total mutations, 15,000 genes that actually have mutations, and about 68 mutations per sample. So that's just a good way to pull out just some general data. First thing we want to do when we have a data frame, we just want to get an idea of what is actually there, right? And the distribution of all the data. So the next thing I want to know is, like I said, the distribution of everything. So I basically took the data file and I basically counted the number of barcodes here to get the mutation count. And then I basically arranged that by mutation count. And that gives us our mutations per sample. And if you check that out, I will show you what that looks like. Mutations per sample, right? Or something. Mutations per sample. So just for every sample, I have a mutation count. It's very similar to what we showed before with this math summary, right? I'm basically making in this table. I could have just pulled this out and squished them together, but I like to use the source data when I do this kind of stuff. Then I just take that file, and I'm basically just doing some basic descriptive statistics. Hey, what is the mean of all those counts? What is the median, standard deviation, something like that, right? And we see, like we saw before, the mean is 68, but importantly, the median is 33, which is a much more important number to look at. So the median, because there's a few samples that probably have a lot of mutations that brought the average up, but the mean, we don't have as many as you think per sample. And we have the minimum, max, the different quartiles, things like that. I want to visualize that here with just a kind of a histogram. And we see the number of mutations on the x-axis, the number of samples on the y, and we see there's a lot squished by 0 that have very few. We kind of go up to the highest. Highest is the average is up around 68, right? And the median, so its average is over here. Median's 33. So here's kind of the median we're seeing here. And there's a sharp tail off here. This is right skewed. Goes down really hard till you don't have very many that have a lot. And if we just look at the first 500 here, sorry, this is the farther one. So you see, we go off really fast. This is just a zoom in to the first 500 here. And so you can get a better idea of the distribution. And here's the density of it as well too, just kind of showing the same thing around 68 or whatever there. So really what you're seeing is this right skew distribution with a long tail of hypermutated samples. That tails off to these samples that have a lot of mutations, but there's not very many of those. So what if we want to know which genes are actually the most mutated? Well, we can do it the easy way and the hard way. The easy way would be to look at this gene sample, and we would see our TP53 is on top, right? And we see the distribution here of which mutations are altered, right? Or we could also do it kind of the hard way, just do it from the very base data, where I just grouped by all of the genes, and I just basically counted the number they are, but I wanted just distinct patients, and then I'm calculating like the frequency and the percentage of them. And then I'm looking at just the top 20 only. So that's going to give us, really for here, I just want really the same thing. I want the mutations, which we have in the other file, but then I also want the samples of mutations, right? So you can see here that 7 patients have multiple TP53s. So you can see 341 have at least one, right? And then some have more than others. And that kind of goes the whole way on that. And we can go a little further. We can break down what those variants are. So are they SNPs? Are they deletions, insertions? So I'm basically just grabbing the data again. It's always the same file. I'm looking at the variant type column, and I'm basically just counting up the number of them. That's all I'm doing. I'm basically just looking at that column, and the BRAF data column, and then just looking. That's all I'm doing in all this, really. I'm looking at that data file. I'm looking at that column, and I'm just like telling you what's in that column. It's really that easy. So I'm looking at that column. I'm just saying, hey, 90% of that column, 62,000 of those counts are SNPs, which you'd expect. Single nucleotide polymorphism, one base pair, right? Then you have your indels, your insertions and deletions. So the thing's gone into the DNA and moved it up, moved the whole code, right? And then these are kind of trivial. And I kind of just made a plot of that. So here's your SNPs, just a GG plot of what that would look like. And then I'm looking now at the variant classification distribution. So remember all these different ones I told you? How will we look at those? So now we're looking in the variant classification column. Same thing as before, same data, different column. I'm just telling you what's in the column. That's it. So if I look at that entire column, I see 56,000 missenses, which is 82.93% of all of them, right? And then you see all these guys here that are the big dogs here that really matter. So that just gives you a good feel for what they look at. And you can plot these yourself, make histograms and all that stuff of all these to get a good idea of what you're looking at. I just kind of want to show you how to find them. We have the boss plot here. You could do more plots too to see the distributions, but I just want to kind of give you just an idea of what we're expecting to see there, right? And one of the things people often like to look at is the tumor mutation burden, right? So this is the total number of mutations per megabase of sequenced DNA. So this is really, really important because it standardized the mutation number. When you have high mutation burden, you have more neoantigens, for example, it can be good and bad, which is better for immunotherapy, but you could have more mutations and bad genes that could be bad, right? Likewise with low TMB, right? So think about it. We already know the mutations, right? We just need to know how much DNA we sequence. And why is this important? Well, think about it. If I sequence, let's say, 10 megabytes of DNA, right? It's not really that much. And if, let's say I find 100 mutations in that 10 megabytes, you're going to be like, wow, that's a lot. 100 mutations in 10 megabytes. And, but the problem is, how do we compare that, right? What if someone else sequences 100 megabytes? That 100 mutations in 100 megabytes is not going to be near as important, right? Like not even near as important, right? So we need to know what you normally sequence. So so really think about it. People normally just sequence the coding regions, right? That's kind of what just the gene regions. You don't sequence all the dark DNA. It's just too expensive now. So we're doing exome sequencing, not whole genome sequencing. So all of TCGA sequencing is exome sequencing. So we need to know how big that exome is. Well, the standard is 32. So the TCGA exome is approximately 38 megabytes. So this is very, very simple. All we do is we take our mutation counts we already know and divide by 38. That's really all you do. So it's the same numbers we saw before. No difference except for dividing by 38. And that normalizes for the exome. So if someone has twice the exome and they have twice the mutations, it would be the same thing. So it's just normalizing for that. And we see here now we have 0.87 per megabyte for our median TMB. And just to kind of look at the distribution of those, once again, very similar to before, we have that sharp right skewed fall off. And TMB high over here, there's not many of them. Here's kind of just a violin plot on it, and you see everybody up here is above the deleterious line, but not deleterious, but high tumor burden, and most of them are below, and a couple are super high. So these guys have hypermutators. They have tons of mutations, right? So let's just compare this to other cancers. What we're seeing here. So there's a good function in math tools called TCGA compare. And if you put in your math file, it will compare the mutation burden to all different cancers in the TCGA. So as you see here, here's ours right here. Here is BRCA right here. So we're pretty close. What's probably happening is we've filtered our file probably different that we've used. We have a few less. We probably filtered out some they don't have. But what you'll see is it's still on the low side, right? All these guys have more. It's still on the low side, which makes sense because breast cancer in itself, invasive breast cancer normally has, a lot of mutations. Here, it's getting all on my face. Let's see. So that just kind of gives you an idea of how they are associated with the other ones. And if you look at categories, I'm going to put these in categories just for the heck of it. So just like we said, like over 10 is kind of high, over 20 is super high, below 5 is going to be pretty low. So I basically just took that category, the TMB data, and I used case when, if you remember. So I'm basically saying whenever TMB is in that column is greater than 5, say low. When it's greater than or equal to 20, say that. Built the categories, summarize them, and here you go on the end. And I basically just made these categories out of the data we already have, no new calculation. And you see here that 96% are in the low category, and just a few are very high, right? Which makes a lot of sense, and is what we'd expect to see in the data. And just for fun for you guys that are a little bit more advanced, if we fit this distribution, this TMB distribution, if you see here I'm using fit dis, which is a really easy way. I want to see how it fits to normal, log normal, gamma, and exponential. I just want to see, how well it, which distribution makes more sense for it, to kind of show you just what it looks like. And what we're seeing here is a log normal, which is exactly what we would expect. So log normal means that a normal is a bell curve. If you log normal, it squishes it over to the left. You kind of lose that negative. So instead of going from negative infinity to positive infinity, you now have a 0. So it squishes that guy over and normally has that right skew on it, which is exactly what we're seeing. And so log normal is being fit to that distribution exactly the way we thought. And, advanced people, you can check this out a little bit better. It's just showing how well that fit. But this is once again showing that it normally does a log normal distribution. So what about those like hypermutators? Well, it was pretty easy to see them. We could just look straight up and they'd be really high, right, on that plot. But if you want to quantitatively do it, you can use the interquartile range to look the IQR method. So what IQR, if you remember back to the stats section in the earlier videos, the IQR is on a box plot is basically the whole box plot. Everything you're seeing is the IQR. It's everything. It's basically a value of basically covering 50% of the data. And you're looking at the quartiles here. We're doing three. If you look at the first quartile, that is the lowest 25% of the data. If you look at Q3 quartile here, it's the highest. I'm studying these. It's the highest 25% of the data. So that's the highest and lowest 50% of the data. If we subtract these two, we're getting the middle. So imagine the middle is the IQR. Everything above that top line is above the Q3. Everything below that bottom line is below Q1. So we want to find everyone really high up here, statistically higher, above the mean, above the median, we can statistically say that this is a hypermutator. So basically, we need to make that upper fence and then just see everything that's above it. That's all it is. So we calculated Q1 and Q3 and the middle, right, the IQR, and we're saying we want the fence to be Q3 plus 1.5 times this middle value. And these can be changed. It's kind of a standard way to do it. And then we're going to look at, hey, filter everybody that's above that fence. So look in the data, the TMB data, tell me every row, every gene that's above, or samples above that fence, right? And then plot it out, and we see that 86 samples are above that. So 86 out of our 990 or whatever are actually hypermutators, and that's like an 8.7% of our cohorts, and they have about 3 mutations per megabyte, which is not even super high, but high for us, right? And we can actually grab these guys specifically, and we see that let's see what I was going to say. Oh, the upper fence was 3. I was saying, well, that's pretty low. The upper fence was 3. So anything above 3 was the fence, right? Because like these guys are much over the fence, right? So check out these, the top 10 highest. Well, 125 per megabyte, that's extremely high. It's 4,700 mutations just in this sample alone. It's crazy, right? So you can see these top 5 or top 6, 1, 2, 3, 4, top 5 are very high mutators. Then we kind of scale down. And even when we get to the 10th one, it's not that bad. But this is how you could kind of find and locate. And all I'm doing is doing data wrangling. I'm not doing any analysis besides calculate the TMB by dividing by 38, right? Everything else is in that graph file. We're just reorganizing this to kind of just to get a better idea of what we see. All right. So now we've looked at our different formatted files, which remember are just different orientations of text data. That's all it is. This happens to be genomic data. It could be any other data. We're just organizing it the way we want, and then we looked at the different distributions of the data, right? But now, since we have that math file, it's specifically designed to use math tools to do really cool, easy publication quality figures. So let's just show you a couple of those. All right. So the core visualizations of math tools. So first, OncoPlot, the most famous plot in all of cancer, right? The waterfall plot. This is what gives you the best summaries. It's usually figure 1 in a lot of papers. It gives you the whole mutational landscape across your whole cohort, which genes are mutated, what genes they are, blah, blah, blah. And do a lot of different ways to be able to show different things. And here's kind of what you're seeing. Here's an example of a really simple one where we have our genes here. I picked the top 20. I'll show you how to do it first. So OncoPlot is the function, and this is the math tools package. We basically just put our graph file, our math file. I want the top 20. You could put all, whatever. And then I want, these are just kind of just some font things. This is the top 20 genes. If I wanted to pick just like these 3 genes, I could do this. Gene equals C for combine, and then just those 3 genes. It would just list those. if I wanted to look at minimum genes mutated in at least a certain number of samples, like I don't want a gene that's just mutated in one sample, for example, I could put a value min mut 10 here. So there's the two ways you could do that. Here's kind of what you get, your basic result. You have your genes on the y-axis, on the x-axis. Here are all your samples. You can list the names as you want. I purposely didn't. You have the TMB on top. And then you have, your mutations. So for this sample, this is kind of hard to see. Just imagine going up, you would grab just mutations in these two guys, right? And then what type of mutations? So you have this legend on the bottom left, tells us all our different mutations. And on the top right, on the right annotation, we see the percentage of mutations. And then we see a percentage of the sample mutated, or actually percentage of the mutations that are TP53, and the types, the number of samples that have. So all these samples have TP53 mutation, but the color codes the different type of TP53 mutation you have. Some are worse than others. And this does a pretty good job. And it's called waterfall because it sorts things to where you see the lesser ones at the end. So there's your plot. Pretty easy, right? Just oncoplot, BRAF, math file, done. So it's a really easy way to do it. And we can customize this, add some more tracks to it. So now I want to add some more colors. I want to change the legend colors, and I want to do a couple of things. I'm going to simulate some data. Since we don't have clinical data, I'm going to add in some. I'm going to add some age and stage data. I think you saw that earlier when I kind of added that on. If we look back at that BRAF data file, BRAF data. So at the end there, I think you remember, you might have seen them. Oh, excuse me. Oh, sorry. Clinical data, right? I'm glad I added this myself, and I show you how I did it. So I added some male, female, and stages. These are synthetic data, it's not real data. But synthetic data is really great for teaching and visualizations. And you see here, I basically just, I simulated this data. I just sampled male, female, 90% female, because that's kind of what you would expect. And then I did stages, and I kind of just made those kind of equal. And I basically just put simulated gender, simulated stage, and then I added these as the gender stage columns in your math file. That's how you see that. So just imagine you have the math file with that, with that data, clinical data column, right? So when you have that, it's extremely simple. Once again, same thing, oncoplot, add your BRC math, but now you go, I want top 10 genes here. I want colors equal to my colors up here. And then I want clinical features. This is where you're adding on your new tracks. So you put clinical features equals C combine, and then just put each one. If I just put gender, that's the only one it would show. If I put gender and stage, we're seeing both of these. I'm also doing I do a couple things here. Let's show you. I sorted by mutation. That gives you a better waterfall effect. I remove non-mutations. If there's no mutations, that sample's thrown out. And then other thing I've done is I've drawn the TI-TV ratio, which is transition transversion ratio, which is also very important at looking at different mutation patterns. And I'll show you that here in a minute, how to calculate that stuff. For now, I'll just show you how to add the track. And look what we got here. We got a bunch of new stuff here. So we still have our 10 genes. We have all our mutations like normal. We have our positions, our distribution of types here. And that came from the same legend, just different colors now. And we have our two new tracks. We have gender along here, and we have age stage along here. And these are by, they're mainly female, and stages kind of change up. And I think they would cluster better if they were real, right? And then we have this mutation. So we have this really cool track here where this is really the mutation patterns for each patient. You have the different transition transversions listed on here. So you have that whole plot there. So you're really getting the mutation patterns, different tracks, clinical features, tumor mutation burden, mutations types. I mean, it's a really, just from a simple little, like a couple of commands. So that's a pretty powerful visualization, I think. So that's it for simpler oncoplots. Now let's look at lollipop plots. And these are really cool plots that basically just show where the mutations occur, like exactly. And it's really crucial to see like the distributions of where these things fall in different functional domains. Now you could do this with packages, but you like to see them. The visualization is really powerful, I think. So, this is kind of what we're looking at here, where we see that this is TP3, and we see different, coding regions, different areas, and we see the actual real mutations plotted along there. And what I've done here, I'll show you how I did that. Lollipop plot in math tools. Same thing, put our math. We choose our gene. And if we want to show mutation rate, show domain label, that's yes or no, it's kind of our idea. We want to label all the famous hotspots, or we can look at specific amino acid positions. If you just want to look at very specific positions, if you want to look, there are just a few different genes here and there, a few areas. You can also do a range like 175:248. And then I'm just kind of tilting them so you can see them better. And one cool thing you can do is you can say C bioreportal equals true. And it will actually tell you what mutation is. So that's really cool, I think. So it gave you a little bit of if you look down here, it told you not only the type of mutation, but also, it gives you the position, the type, and the classification. Now, we already know those, but it's kind of cool to see them again there, I think, right after. And we kind of see here, like, a lot of these mutations are really associated in this, DNA binding domain. So we could expect to see in an oncogene like that. So yeah, and you can filter this out. You can filter less genes in your math. Or you can choose specific genes in your math, kind of reorient that to get, if you just want to show a couple here or there. Just wanted to kind of show you how to do everything. We can do the same thing with PIK3CA. Same exact stuff. I'm just putting PIK3CA up here and showed you everything else the same. And you can see in this PIK3CA I just said that TP3 is a tumor suppressor. PIK3CA is an oncogene. And, in oncogenes, you expect to see, a lot of different kind of transitional domains that are important. And you're going to see that these mutations really flock to those areas, right? And you can, and by adding that CO bioportal link, we got a good definitions of protein changes and classifications. So that's really cool. Like I said, you could read this to show that we're seeing classical patterns in there. And one more thing we're going to look at, kind of comparing different ones. Remember the lobular breast cancer one, CDH1? Let's take a look at that. Man, it has mutations everywhere. But, if we looked at probably some of the more important ones, like the truncating ones, we would see, some of these early ones. We see the early positions here. So these are found right here. But I'm just showing you another way of being able to visualize this data in lollipop form, which I really love. Lollipop plots, they're really cool, and paper people love them. One other thing you look at is that, remember we talked about that transition to transversion analysis? So really this tells us our mutational fingerprint. So different mutational processes leave different fingerprints, and these often are associated with different changes, pathways, things like that. So having the mutations is a good way to start early on to start recognizing these patterns. And as you guys probably already know, the genome is made up of 4 different base pairs, A, T, C, G, right? Guanine, adenine, all of those guys. And if you remember, AT linked together and CG linked together. And if you remember too that there's 2 different structures of genes. So A and T are a purine and a pyrimidine, right? They're different structures, and that's how they chemically fit together. So you could imagine if a mutation here is a purine and it switches and stays a purine, then it's going to be a good match still, and we'll be able to do well, and we'll probably be able to make that protein. That's a transition. We transition, right? If the pyrimine turns into a pyrimidine, the mutation changes the structure of the protein. That's going to change everything up, and the DNA have to adjust to be able to come together and stay together. So a transversion is much more likely to be dangerous. And so you're normally going to see a higher number of transitions than transversions, and the higher the number, the better. The higher the ratio, the better. The lower the ratio would mean there's more transversions, right? So the higher the TI-TBA ratio, the better for the patient. But really, the structure of the mutation patterns is really what tells you the difference. So you see, like, from C to T, for example, is a very common mutation for everybody. The most mutations are always there. Then you see the different ones that have been changed, and you only see 6 here. You're like, well, how could there be 6? Shouldn't there be 12 different type of changes if you have 4? And you're thinking, yeah, that is true. However, there's a standard of classification. So we talked about the purines, right? If you go purine to purine, sorry, I should have said it's not A to T, but ATCG match, but it's the purine to pyridine match, right? So really, it's purines A to G and pyridine C to T, if that makes sense. What's really to know is it's just that when you have a purine to purine, it's a transition, not that big of a deal. But if you have a purine to pyrimidine, it's a bigger switch, and it's gonna be worse, right? So a typical ratio is around 2 to 3 for most cancers, but if you have deviations, that's the point of tackling these things. You might have really interesting mutational processes and a lot of different interesting pathways probably altered. So, and one way to look at that, let's see, where did I have it? All right, we'll probably look at that later. I'll tell you a little bit more about this in the below in the practice problem. And after I told you to do all that stuff, all you really had to do was use plot math summary. So if you do plot math summary in your math, you can remove outliers, and here you go. It has everything you need, right? You got your variant classification, variant type, your different classes, your variant class summary, the histogram of mutations, this guy you saw in the aqua plot. This is a really great, just super quick summary, a dashboard basically of a lot of your descriptive statistics. So I wanted to show you kind of at home do it manually, because you can apply that to any kind of data, right? But you can also use this map tool function to get this quick summary of it, and it's just a cool way of getting all of that data really good, right? You can also look at the variant and low frequency. So what this is let's say you have 10 reads at a site. So that means we've sequenced 10 different reads at one DNA base pair site, okay? If every single one of those is found to be, the same allele, that's your reference, right? So if all 10 are referenced, then you don't have any problems. There hasn't been a mutation. If there's one of those is an alternate allele, then that could be a problem as well too. But it's a very low allele frequency. That would only be a 10% allele frequency, 1 out of 10. If 9 out of 10 are all alternate alleles, that's a 90% allele frequency. The higher the allele frequency, the worse the cancer type is going to be. So that's going to be very important in figuring that out. And our BRAF data actually has it. So if you look at the column, the VAF column, that actually tells us exactly what it is. But, however, that's just, we can calculate it ourselves as well too. So remember, once again, the math file is going to give us that. We can look at it. But we also know the T reference count and the alternate allele count, right? So if we divide the alternate allele count by the reference count, we'll also get the allele frequency, the variant allele frequency is what it really is. So here, all I've done, don't worry too much about it, is I've divided those two things. You see here, alt count by numeric count, right? Better get low. And what you're seeing here is that distribution, a distribution of those variant alleles. So we get a kind of an idea of what that looks like. And just kind of putting this all back together into one big summary, we see, once again, we have our total samples, we have our tumor TMB that we talked about, we have our top 5 genes, and they're what we'd expected. It's pretty cool, right? 341. The most mutations are in TP3 individual, but the most total were PIK3CA, which is exactly what we thought, right? PIK3CA first, PIK3 second, and CH1 kind of pulling up the rear pretty close. So we got exactly what we expected from the data, right? But we mostly had missense mutations where stems predominate, which is also what we'd expect. And our key biological insights, just like we talked about, PIK3CA is most frequently mutated, it's oncogene, TP3 are kind of subtype dependent. If you wouldn't look back at some of the other data, we could look at that and kind of show that a little better. We see that TMB breast cancer has a pretty low TMB. And the mutation spectrum shows a lot of different processes with C and T that is dominating, right? And if you look below here, we look at our quick reference guide here. And this gives us a view, just kind of a basic idea of how you load your math data, and also how you query it and how I made the math file. It's just a really simplified version of what I had below. Here are some basic tools, exactly the tools I showed you above. There's one more showing the Get clinical data summary. That's just one more way you could summarize it. And there's the TMB calculation, the variant classifications, just a bunch of that stuff that we would expect there on that. So now let's go through a few of these practice problems here at the end, just to kind of give you a deeper understanding of what is going on with everything here. Okay, so first we got basically GATA3, right? Let's look into GATA3. We know this is a transcription factor that plays a very crucial role. It's a luminal marker associated with ER, positive breast cancer, and it's very frequently mutated breast cancer. It also often has frame shift mutations that create truncated mutations. So this is a very, very important gene. It's very well worth looking more into what's going on. And let's see here. So we have, so let's look at part A first here. And let's see, excuse me one second here. So first thing we're gonna do is calculate the frequency of GATA mutations. So I'm gonna look at, let's look at, sorry, I'm just organizing it right here. Let's look at our BRAF data and filter for just our GATA3 gene. So we want to look at the data just like before. I'm just looking in the column. And so we're looking at the BRAF data, and we're filtering by the GATA3 gene only, and we're looking at the tumor samples that are unique, right? Just the unique. So we just want all the unique samples that have GATA3 genes, right? And kind of like before, we just looked at the unique barcodes to get the number of patients, and then we're dividing the mutated samples by the total samples to calculate just the frequency of mutation, right? We're plotting it out. We see we have 125 total mutations, and in GATA, out of 990 samples in the cohort, so 12.6% mutation frequency. So we see these are unique samples, right? So we see that as well. We could also use the math tools function here, where we can look to the Get gene summary and just kind of look at GATA gene only. And you see here we have the number of mutated samples and total mutations. So two samples have more than one, obviously. And this kind of tells us what we expect. It's found in about 12% of cancers, and the biological context of that is it's a master regulator of luminal cell differentiation. We can create the lollipop plot. So remember, just put the BRCA math in, do GATA3 only if I don't open up crazy windows, and look what we got. So we just have the different hotspot mutations for GATA3 here, and we can kind of look into those more at a different time. I pulled those out. So I'm looking in the data for that gene, and I'm basically grabbing the columns that I want and organizing them. And I'm basically just seeing that the highest number of occurrences of GATA are normally splice site mutations affecting this protein, and so on forth. You can look at the different ones. Then I'll kind of just give this, here's a little interpretation of the lollipop. It just kind of shows you that there's a lot of mutations in the frame shift area, for instance, and most frequency mutations are in frame shift in certain hotspots, things like that. It also has a little bit dome structure thing, but you can kind of check this out later. It's basically just showing the biological interpretation of these different things. And for part C, we have the variant classification distribution. So once again, I just grab the mutation data, I filter out that for that specific gene, and I look in that variant classification column. So I'm just looking for that gene only those rows, look just in that column, and then just count out everything, just like before, and just kind of see what is actually available for that here. Let's see here, sorry. I'm trying to pause, my computer's dying. This is a live version for sure, where I have to go grab my charger, right? So we're gonna do a distribution of just that here, and you see just the distribution of those different variants there. And just like we saw before, we just want to see the distribution of the variance. No different than we had before. And I kind of found the position, kind of the percentages here. Same thing. I just organized it a little bit better and just kind of showed what is happening in GATA only and what is happening overall. So you're seeing this is a big deal. There's 48% of GATA mutations happen to be frameshift insertions, where normally there's only 1.4% of those are like that. So man, these are dangerous mutations. All three of these two indels and the split are really dangerous mutations. So the fact that GATA3, so anybody that has a GATA3 mutation, it's a very dangerous mutation. So it's very enriched and being deleterious and being GATA3. One more thing we can look at is the tumor mutation burden in GATA3 and look at it and compare the mutated versus the wild type. So we're just going to do the same thing. Remember, we divide mutation count by 38, grab just the GATAs only this time, and we see here, we have a little descriptions of those, and we see that the median is 0.68, and the mutated, and 0.89 in the wild type. So it's lower. We thought we wanted it better or be higher. Well, kind of. It matters what, right? It matters the therapy and the cancer, but as you would expect in breast cancer, it is on the lower end. And if we compare these, the plot, we'll see these are kind of on the lower end. And if we do an actual Wilcox test on that, I'll leave it to you to kind of check that out. It's just what I'm doing, just like I did it in the statistics things last semester. We got a significant p-value, and that basically tells us that there is a significance, that the wild type is significantly lower. So that's really good news and what we do expect in that cancer. Now, give me 1 second. I'll have to edit this later. I have to grab my chart. All right, you can tell this is live. I am not AI. Because darn, this is a long presentation. I had to grab that, sorry. It's crazy how fast this thing's going off lately. All right, so we're doing great. So now we've really shown that not only can we do the same analysis, but we can just basically filter GATA3 along the way. And I basically just reproduced almost kind of exactly what we did before, right? I just took one more step of filtering out the rows that just had GATA in only, right? And then we kind of apply the same different things to get our basic, our basic, statistics of mutations. These are all things we did earlier, right? We just took it one more step to look at GATA only, right? So that's basically the only difference in that data, right? So let's move on to the second problem here. So this is just a more in-depth, just taking one gene and going more in-depth. Let's see here. And like the lollipop plot wouldn't make sense for one gene, right? So you really wouldn't have to do that. You would maybe just do a box plot or show things like that. All right. So let's look at problem 2 now. Exploring signatures preview. So these last two are pretty advanced. So if you get lost going forward, you're a beginner, do not worry about it. I do not expect you to be like, oh, this is really easy. This is for people that have more of a genomics background. Or when you work up and build up your own background, you can just check back to this and be like, oh, well, dang, that's really cool. So let's just dive. I want to dive more into looking at those mutation signatures, because that's something that people are often going to want to know about when you have mutation data. The first thing they're asking you is like, what is the pathways they're involved with? What are the mutation patterns? Are there signatures? So this is something you need to really look into. So let's first look at the different substitution types, the basic building blocks of mutational signatures, so you'll have a better understanding of what is happening, right? So first, let's just look at SNPs. That's all we need to look at for this. So once again, we pick up the data file, and we filter for just SNPs. We just want the rows that have SNPs. So this gives us 62,000 SNPs, a lot, right? So we just have our SNP data, and I'm basically, I'm just making it kind of simplified. If you see a C, greater than sign, I'm just making it a C. So I'm just like simplifying things here for you. And I'm just standardized this basically so So basically what I'm doing, 1, 2, 3, just so you don't get confused. I'm basically referencing these to the standardized thing. Sorry about that. I forgot this comes after. So basically, by convention, we expect mutations from C or T, not G or A. This is because C of T is the complement of G or A, right? On the opposite side of the DNA. So if you have one, you have the other, right? So by just convention, you always switch back to C or T. So if you're A, you go back to T. If you're G, you go back to C. So basically all I've done is do that to try to standardize all the different subtypes. So I just basically rename things, standardize them, and honestly, you could just steal this data and copy it and use it another time. Always, if you see something like that, that's a little bit complicated, there's a basic thing that's going to be used on basic files, just take my code and reuse it. If that's one thing I could teach you, is it's okay to take code and recopy it. That's how you learn. That's how you get things done. You can't just spend all your time learning everything. Then I basically just counted. I want to know how many C to Ts, how many things like that. So all I've done is just rename things in the new standardization. I plotted them out, and now you see that here's the frequency of those 6 substitution types. So anything from C, you see, remember how we did that plot before, and C of T was by far the highest? We're just thinking the same thing here. This is what we expect. That transition, super high counts. But now, we have 6 subtypes that kind of show everything. And this kind of shows that too here, that we always want the pyrimidine, the C of T reference, right? So if it's G to A, we always change that to the complement C of T. If it's A of G, we change it to C of C. So that's why that's what I'm telling you about how you change to the complement strand, just to standardize them all, because they're basically the same thing. And it just makes it a lot easier to look at a later time, right? So let's look at the distribution of all those things. It's just the distribution of the data frame I just showed you. So once again, showing C of T as the highest. So this just kind of showed you pretty much what we did above, but just kind of more how we did that, right? And let's just calculate just C of T only. And like 43% we saw earlier, it's just kind of showing you some different ways if you wanted to calculate it yourself, a different way. And this is kind of interesting to do. So if mutations were random, there's 6 types, right? Each of the 6 types would be 16%. So we would expect mutation on average to be that type to be seen 16%, 7% of the time, right? So it's expected to be seen 16% of the time. We actually observed it by 43. So there's almost a 2.6 times enrichment in those and CFTs in this sample, right? So we're really seeing this is way above random. And let's look more into the actual distribution of transitions, transversions. And we see, as expected, a higher number of transitions. And we calculate that ratio is 1.09, which is pretty low. The typical range is 2 to 3. So we're getting more. We're getting less transfer. If we had more transversions, the number's lower, right? So this is actually worse. A higher number is actually better. So if you have less transversions, it'd be higher, right? So this is actually, we have more transversions than normal for the cancer. And this is just kind of telling you a little bit about why do we have so many CFTs. It has a lot to do with these CPG islands. If you have a lot of CPG sites or islands, you'll see a lot of those mutations. I'll kind of leave that to you, but it's basically giving you kind of the aging signature there, just de-animation. But you can please look at that, read a little bit more about it. This talks about a little bit about that too. A little background of why those are important and why we see those a lot at the time. So yeah, the key takeaway is we're seeing primarily CFT transitions at CPG sites, and this is just spontaneous de-animation. We often see these. I would say that if you have too many CPG sites, you could have some sequencing errors too, something to think about in the back of your mind. So let's now look at the last problem, building a custom oncoplot. So far, if you look back at the videos last semester, I used FeatMap a lot for plotting, and we used oncoplot through the math tools to plot your oncoplot. But going forward, we're really going to try to move toward using complex heat map. This is a package in Bioconductor that was originally made just to make simple heat maps, simple hierarchical clustering, and now they're adding more stuff like oncoprints, other things, and it's just a very well-maintained package for the last 10, I've been using it for 10 years. It's pretty much what everyone I know uses to do heat maps. And now that they do oncoplots, I'm pretty sure everyone's using it for oncoplots too. It's just a little bit harder to use at the beginning. And that's a good thing because there's so many different features or things you can do, and it's just completely open box. You can just change any part, any color, any track, anything. And once you learn how to use it, it's really not that hard. I'm being honest with you. Once you get it, you'll never have to learn again. And you can be using this thing for all of your data. It's not just this math tool data. This is the last thing you use it for. Any kind of data in a matrix, in an X-Files file, you can use complex heat map. That's actually the better form for using it. So one of the things we're going to have to do is we're going to have to take this math format and change it into a way that's actually useful for complex heat map. And what complex heat map needs is it wants all the rows to be gene names and all the columns to be sample names. It's kind of right, and then it wants more columns for annotation that are just like are above. So imagine you have your genes. You have your samples on the across, and then for each gene, you have another column that has like what kind of mutation, what's the CIF score, whatever you want, whatever clinical data or annotation you want. So let me show you how to actually build that file. So you take that BRAF data file, and I'm going to create a tibble, which is the data frame, and then I'm going to select just these 3 columns. So only ones we need for this, because we just want gene names and sample names, and basically just mutation type for now. Because we're only doing one track for now. And we pull that together and we see that we have, this mutation, we have this data frame right here now. So we have our first column is the actual gene name. The second column is our barcode. And you see there's multiple, these are not unique, right? These are all the genes for this barcode. And then we see the variant classification. That's our next column. And let's just kind of look at that. We can do our count on these column, and that will kind of give us our the percentages and numbers. We already had those from earlier, just a repeat of what we did earlier. And we can also simplify the mutation types. For example, we had frame shift deletion, frame shift insertion. Those are really just indels. So we'll just make that into one. We'll just call it just one thing, indels. So we put it through a few of those together just to kind of simplify it, and now we have in frame indel instead of the different ones. So 1, 2, 3, 4, 5, 6, 7. We took a couple away just to kind of simplify the categories that could be coupled together, right? So we've just simplified the categories. We transferred the data a little bit or transformed it. We've simplified the categories. And then now we're going to select just the topmost 15 genes. That's all I'm doing. I'm just taking our data. I want to take the unique out of everything unique out of the gene names tumor sample. So I just want, one thing for each. Then I want to count those. And then I'm going to, now that I have the count, I'll be able to find the top, the frequency. Then if I know the frequency, I'll know the top 15. So really, it's not that complicated. All I'm doing is just making a frequency and then taking the 15 most highly mutated genes. So now I have the same matrix, except now it's kind of different here. I have the genes here and then just the frequency, and then I know the top 15 that I can select there, right? And now we're going to create that final mutation matrix now that we know the genes, right? So the top genes, I called this, remember up here, top genes. So top genes contains those genes already. If you went over here and you looked at top genes, you would see top genes here. Chromosome 15, TP3 number 1, PIK3CA second. Just a list or a vector of names, right? And I basically have that mutation data. Like the first one we transformed. I'm just going to filter for just those genes. So I want this matrix with just those genes. And then this goes back to reshape. If you remember back in the third boot camp, we had that pivot to wider, longer. So if you remember the genes here, everything needed to be, remember the sample names went vertical, right? Well, we need to change those and pivot them out so they're actually the column names. So that's what we're doing here. We're making a long format. So we're just taking our mutation data with just our top genes. We're grouping by the gene signal and tumor barcode, and we are kind of just basically fixing that. And then we're flipping it here on wide format, which you run pivot wider and basically just list the names that they were called and then what the names they should be. And you can read more about that in the bootcamp. And we're basically just reorienting the matrix how we would want it to be. And now we've created it, and let's go look at what that looks like. Mutation matrix is the final one, right? Mutation matrix. Here we go. So perfect. It's exactly what we want. So this is kind of what you want for, and this might be what you are more accustomed to as well too. Is look, the gene names are the row names, and that's a really good thing too. See how I did the column to row names? Remember the second day at the second boot camp, I did the column to row names, so I was able to move the gene column over to make it the row column, right? So that's cool. So that's how the genes are here, and then the columns are each sample. So for each sample, you can go down each gene, and it'll tell you all the different mutations in here, or the top 15, right? So this is frame chip, mid-sense, frame chip. Obviously, these blank ones mean there's no mutation there. So now we have this kind of, you can already kind of see it, right? A heat map. So now we can plot this heat map out. We don't even have to do oncoplots. We could just plot this itself. It would look cool, right? We'll worry about that next time. Because you can plot and cluster this and find different clusters of genes and stuff too. That's definitely one of the next steps. But let's first just make the oncoplot. What's cool is you have it in the format now. And if you have this format, you can do complex heat map stuff. You can do anything. You can do complex heat map. I like how everything's reversed. How do you fix things when they're reversed? You can do all the clustering, the analysis of the complex heat map, and you can also do your oncoplot, all from the same matrix. So this is a really good matrix, and sometimes this is the last matrix you get, right? Or the only matrix you get. So you could actually start here too. We'll talk more about that later, a different workshop. But anyway, so I have my matrix. So now I feel good about it. I can actually do my oncoplot. So let's just understand how complex heat map works now. First, I'm going to define my colors. So I want to do my legends, what colors everything should be. So I just have all my names. So I have basically a vector, you can call it mutation colors, of all the colors. Two, so if you remember, oncoplot has basically just tiny little rectangles, right, that told the right color of mutation or whatever. Green was missense, whatever, right? Well, we need to tell complex heat map more information than oncoplot did. We want to tell it, we want a rectangle. They could be circles if we wanted. They could be anything we want. But I'm going to make them kind of a standardized thing, how you normally see it. And I know this looks very scary, but you only have to do it once. And this is kind of a template, so you don't have to really change it. And later when I'll show you the link to the complex heat map site for the tutorials, you'll see that it's not that as hard as you think. It stays the same, and you just tweak things a little bit each time you do a plot, right? So what's basically I'm doing is I'm creating the background and the grid, things like that. So the first thing I have to do is create the background. So this function, all this will always be the same. This x, y, y, wh, width, height. So I just want to do the standard ones, but I want to multiply the width and height by 0.9. I want to make them a little bit smaller. And then I want to add this color, all right, which is going to be pretty much light gray on the back. And then for each one of my guys here, I need to tell what color and what shape, right? So for missense, same stuff. I want 0.85, 0.85, and I want the color from missense, right? Same thing with nonsense, same thing with frame shift. By convention, indels are a little bit smaller in height. They're half the height. That's why I put 0.5 here. You don't have to. And so basically all it's doing is just setting the width and the height for each thing and changing the color. There's nothing else happening here. And then once you if you only had missense, nonsense only, you would get rid of all this stuff. And if you want to change the width and the height, that's fine. Colors, anything. Once this is set, you're done. You don't ever have to do this again. And really, you don't ever have to do it ever again if you're doing, when you change to your new data, just change the different mutations you have, mutation types you have, leave everything else the same. You'll always have this alter fun vector, okay? Now, once that's set, it gets a lot easier. Let's just make a simple plot here. So what do? OncoPrint is this. So we call complex heat map package the library, and OncoPrints the function. We put in that mutation matrix we made. The alter fun equals that alter fun vector we made, and we put the mutation colors. For now, top and right annotation are null, they're empty. I want all the row names on the left. You could put right. Here I just made the font size. I don't want to show column names. I do want to show position on the percentage mutations on the right, just like before. Font size if you want to change it. Border around cells, yes or no, color, and then just the title. And then you write, for complex heat map, you always have to make an object and then draw the object. So I'm just drawing that object. And look, very similar to before. We have our genes, and we purposely picked out the top 15. We have our samples here. We have our legend that shows our different types, right? And we have all of our genes here. And, our different mutation types, percentage of mutations. Not bad, right? So we got all that stuff going, really great. But how can we do more? What if you want to add annotations? Remember the tumor, the TMB, the age, all that stuff? It's really not as hard as you think. So basically, I just had add more columns, right? Add more columns to mutation matrix. I want to add a column. I basically took the mutation count divided by 38, added that as a column called TMB. Now we have the clinical data, the tumor data. Then I made some more simulated data, and I added the cancer type, the 5 different sample types. I added a column called subtype. I made a new age and stage. So we have age, stage, subtype, TMB, all synthetic data. All I've done here is taken the mutation matrix that we started with, and obviously I made a new data frame called clinical data, and I added all these variants in. So you can either add it on the end, append it to the end of your mutation matrix, or you could add it as a whole separate matrix. It just matters which one you call, right? So I basically just have a new data frame. The rows are the sample barcodes, and then the columns are TMB, age, stage, those things, right? You see here kind of what you're seeing. You see the sample number, total mutations, the TMB, the subtype, age, stage. This is kind of our clinical data annotations, right? And now we're going to kind of make some colors for those things. So we add subtype colors, we add stage colors, and then let's build that together. Let's build a new thing, right? So build the top annotation object. So that basic heat map we already made, that stays the same. We just basically add in, remember when we said top annotation equals null? That's all we're changing. So that the backbone, the framework is still there. So we basically want to build the top annotation object first, right? And to do that, the first thing we do is we want to ensure the clinical data is the same order. So this is just checks, right? We want to make sure the clinical data and the mutation matrix match. So we just kind of check that, and then basically we make our object here, where we write top annotation. We call it heat map annotation, and we want an annotation bar plot. That bar plot on top with the TMB column. So we're just doing heat map annotation. We want to call it the annotation, call it TMB, and name that annotation bar plot, call that function, and then just call that column in the data, and then just kind of what color do we need it and all that stuff, right? And if you want to do another one called subtype, we just use subtype. If you want to, basically just calling each of these columns, right? And then we're throwing in our colors. And then we just tell it where we want it. We want it on the left side, and then here how big it is, kind of gap between, and some legends, right? So really all we're doing is we made our data frame that has different annotations. We made colors for them, and then we just called each column differently. And if we just called one, there'd just be one new one. And basically we're expecting all these to be top annotations, and we told it where it was, things like that. And then we actually create it, right? So it's the same before. We have OncoPrint, we have our mutation matrix, we have our alter fun, we have our mutation colors, And then here's the difference. Top annotation equals top annotation. We've changed nothing. Everything else is the same except top annotation equals top annotation. And then bam, look what we got. Beautiful, right? So we got our waterfall plot as expected. We have our TMB on top, and we have our tracks on top, which looks way better than the bottom. So math tools can only put them on the bottom, which is really annoying. I think they look better on the top, and you have much cleaner legends here. And you can do a lot of things to the legends too as well if you want to have different shapes and put them different places. Well, how do I add a side annotation? So this is more like sample level, right? Because these are all the samples. These are sample level annotations. Well, I want to add those side ones. Remember how we had all that stuff on the side? How do we add those? Well, first, let's make our gene pathway is basically just another data frame. We'll grab our top genes, and we're basically saying when we see these genes, we're going to call it this pathway. So case when. Case when the genes are these, we call it that. Cases where it's this, call it that, right? So that just adds in all those, right? And then we call these tumor suppressors, these oncogenes, and then here's what we got. We basically just took data we already had and just put it all into a new table, an annotation table. Where, remember before we had the sample names and annotations, now we have gene names and annotations, where annotations are the pathway or our gene type. They could be anything. And then we want to, once again, add the colors for those annotations. And we'll just kind of calculate some mutation types. It's not so important. Here's the important thing, the row annotation. So heat map annotation function is on the top. Row annotation function is on the sides. So we're basically saying mutation count, just like before, is a new track. And we do the same thing, NO bar plot. And we just call the gene mutation counts. And if you look above, what that really is, the counts we called. That's why we made those counts, right? Gene mutation counts. And it's basically giving an idea of those counts. Remember how we wanted the percentages, right? So the number of mutations, right? And then we add in a pathway and a type where it's just calling those columns once again. And then we add the colors, and then we throw that on there, and look what we see. We have tons of the tracks now. We have all the tracks on the top as before, but now look at the right. We've added several different things here. We've added the frequency of mutations along there. The frequency and number of mutations has been added, and we've also added the pathways and the type. So we have pathways here. You can see the different ones, and then we can see the different subtypes as well, different types of mutations as well too. And these could be anything. Any kind of things you want on the right, you would just put as an extra column on that annotation file, right? And you can also do things like, so that's really anything. You just make two different data frames and do a top annotation or the right annotation. One thing we can also do is split up the heat map, which I think is really cool. You can do that to group samples by different things, by molecular subtype, genes. You can highlight specific patterns. What I'm gonna do is I'm gonna split it by the different types of breast cancer, different kind of subtypes. So I basically took that column that we already have, and I just made them into factors, and I just added that on to. So we already have our top annotation and our row annotation, and what I have done here. Let's see where sample split. Here we go. Right here. Sorry, I got lost for a second. How do we want to split? By the samples. So all we've done is add this extra thing in. Everything else is the same. We just do sample split, which should be factors, for column split, and just how big of split do you need. And look, we have these splits now. So these are the 5 subtypes, and we have a split between them, and the waterfall is organized everything within each split, which is really cool. So we get a lot more. We can do more splits. We can split by the pathways here. So this time I did a row split. So I factored out the different pathways, and I made a row split. And that's where we got these guys as well. So you see all the rows are split up. So you can do either way. You can split both ways. There's lots of different ways you can do it. You can also customize the legends. You can see here, you can just add a lot of different features to the legends. And I'll let you kind of look through that on your own, but you can kind of add new colors, shapes, and organize them in a better way that looks a lot better, right? You can also just grab for one gene. So at the very beginning, we could have filtered just for TP53, just pulled distinct barcodes for P53 only, right? So I only care about that only. So I'll just kind of do the same thing as above, but I just kind of selected for that, and now you can see they're split between the two different types. It's really nice. And here's just kind of showing you how you can save that in different shapes and things like that to create your final plot. And finally, just kind of a summary of some of that stuff we talked about, of how to do some of those things, and some just kind of key takeaways for you. One last bonus challenge you could do with math tools is there's a function called somatic interactions. It's basically just doing a correlation plots for you. So why not? You can use core plot, you can use core, you can use this. So here, all you gotta do is put your math file in, name your genes, name your significance thresholds, and bam, here we go. And it's basically telling you here, like the co-occurrence, how much do these genes co-occur? So for example, TP53 and PIK3CA are almost totally opposite. If you saw earlier in the plots, whenever there's one, there's not the other. It's very rare because they're both affecting a similar pathway, so they only need one or the other. And if you look here, it's highly significant that they're mutually exclusive. So it's exactly what we would expect from this. So this is a good plot to show you how much things occur, but also how the significance, are they actually significant? And you can kind of pull out, different data from that to see like how many are mutated, things like that. That's I'm just kind of reorganizing and data cleaning. When there's something like this, I'm just data cleaning and organizing. And I know that kind of looks hard, but it's something you'll get better at, and AI can also help you with. And also, I just want to do a really quick check to see if that actually is significant. And once again, I look between those two, and it's extremely significant. So that's really good. And it's to be what we'd expected. And this is just kind of looking at some of the co-occurrences between those different things. And I'll let you guys kind of look more into the biology interpretation if you would like. And here's just some of the things we learned, during going through the problems. And also kind of give you some additional resources to look at. TGJ Data Portal, CBio Portal, Cosmic are places where you can look into this data we downloaded, find some of those math files, things like that. You can learn more about math tools here, and learn more about SNPs here. For the cheat sheets, like we did before last semester, the tidy versus ggplot 2, you'd always look at. And here's that complex heat map tutorial that I wanted to show you. There's a really good tutorial here. It starts at the very beginning. Here are just, heat maps, just normal heat maps and things. And here's the OncoPrint setting for you. You see how they go into really looking at how do you do this all different ways. There's so many different things you can do with these OncoPrints. But you really look into how that is working from that. That's a really good, I would really suggest you go through this complex heat map tutorial. It's going to take a while, but the more you learn about that, the more you'll use it in a lot of different of your data sets. So that's going to be it for today. So preparing for the next workshop, we're going to be going kind of further and building upon everything we did here today, kind of look more into these significantly mutated genes, learn more about the hypermutated phenotypes. We're going to actually cluster things and look at different things like that. That's going to be really cool. We can do some pathway analysis, some survival analysis. And we can also look at some actionable mutations that actually might be linked to drugs that might be able to help them. So that'll be really cool. I'm looking forward to that. And I hope you guys learned a lot from this first workshop on DNA. And we're using RNA DNA for analysis. And I will see you for the next session soon. Take care.